Hello friends, how are you? Welcome to the Japan Study Center. Today we are going to see the chapter of science and technology. And what is our topic today? Today we discuss one most important chapter here, heredity and evolution. That is a class 10 CBSE ncert science and technology book here. That's important chapter. But here question arrive here. What is a DNA and function? What is a DNA? And the right the function. That's important question here. DNA and function first of all here. And first of all you understand what is the full form of DNA. You know that the full form of DNA here deoxyribonucleic acid. Deoxyribonucleic acid is a polymer composed of two polynucleotide chain that coil around each other to form a double helix. The polymer carries the genetic instructions for the development and functioning, growth and reproduction of all now organism and many virus. DNA is a ribonucleic acid or nucleic acid also here. You know that. I repeat again, the DNA here, or DNA is full form here, deoxyribonucleic acid is a polymer composed of two polynucleotide chain that coil around each other, and also here a double helix. And here important point here, the polymer carried genetic instruction for the development, functioning, growth and reproduction of all the organism and many virus. That's important here. And also important here, show this picture here. You know that in this picture there is a helical structure of DNA here. The clear, they are joined to each other. There is a most important here, a four part here. There is a four part here. There is a adenine, thymine and guanine and cytosine. Mean A attached to here T and G attached to the C. That's important here. There is important. First of all, see to the upper side here. You know that here a pink color. There is a represent adenine and attached by here thymines. That's important. And other part is here second number. They are attached by the guanine and they are attached by the cytosine. That's important point here. There is attached by the AT and GC. Note that this important helical structure for the exam view here. And that's important picture here. And here nucleic acid, the organic component known as the nucleic acid, and can be found in the form of DNA or RNA in all living things. That's important here. There is a DNA and RNA in all living things present here. And this nitrogenesis basis, you know that this is a nitrogenesis basis. Sugar molecules and phosphate group are the joined by the various bond in a series of sequence to generate this nucleic acid. The DNA structure define the basic genetic makeup of our body. That's important here. The DNA structure define the basic genetic makeup of our body. And DNA is responsible for the carrying and transmitting to the heredity material or genetic instruction from parent to offspring. That's the most important here. In addition to be ensuring that all living things pass on their genetic information. That's an important function here. In addition to ensuring that all living things pass on their genetic information, and DNA is essential for the synthesis of protein. The DNA here, a synthesis of protein. A nuclear DNA is the DNA that is found in the nucleus of each eukaryotic organism cells here. That's important point here. The majority of organisms genomes are coded for by it. with the remaining genomes began to be handled by the plastocyte and mitochondria DNA. And the mitochondria DNA refer to the DNA that is found in the cell's mitochondria. And also here, similar to this, plastocyte contain their own DNA and the crucial for the photosynthesis. That's important function here. Know that the important function on your screen. But next question arrive here. That's important. What is the RNA? And write the structure function of RNA. You know that today discuss for here RNA structure and function. One by one explain on this topic. First of all here, what is a RNA? You know that RNA is important and full form is mostly important here. The full form is RNA is a ribonucleic acid. Ribonucleic acid that help in the synthesis of protein. There is synthesis of the protein in our body. You know that RNA is most important structure here. They help in the synthesis of protein in our body. And this nucleic acid is responsible for a production of new cells in the human body. That's important. This nucleic acid is responsible for the production of new cells in human body. It is usually obtained from the DNA molecules. That's important. It is usually obtained the DNA molecules. RNA is a part of DNA. Show their picture. This clear your mind. But next, RNA resemble the same as that of DNA. RNA resemble the same as that of DNA. The only difference began that it has a single standard, unlike the DNA which has two standards, and it is a consist on the only single ribose sugar molecules in it. That's important. That's consist on only single ribose sugar molecules in it, or hence it's a name of ribonucleic acid. RNA is also referred to as an enzyme, as its help in the process of chemical reaction in the body. Know that. Let's see diagram here. You know that on your screen there is a most important here. One side of DNA structure and other side is RNA. You understand there is a part of different, but I remember that there is a part of same here. The RNA is here ribonucleic acid is atom 9 uracil and cytosine and guanine. That's important part here. But DNA here, deoxyribonucleic acid, they're attached with here or thylamines, atom 9s and guanines and cytosine. That's attached here. You know that of helical structure on your screen, that is attached by the CGAT. But other side RNA only single. There is AUG. That is single. Why the single? Let's see other picture here. On your screen, there is a most important picture here. You know that. Clear your mind. The DNA here, RNA here part of DNA. You know that they are attached to each other, but upper side is separate. The DNA has the same but two helical structure, but other side is one part of RNA. That's a part of DNA. That's important picture here. And also there's a replication. There are DNA and RNA translation of the protein. There is a replication. Replication is the most important here. Replication here, transcription and is of transcriptions here, a translation here of the protein. The RNA DNA is make a protein. That's important diagram for here. Let's see function of RNA. The ribonucleic acid RNA, which are mainly composed of nucleic acid, are involved in the variety of function within the cells and are found in all living organisms, including bacteria, virus, plants, and animal. You know that is important here. The variety of function within the cells and found in all living organisms. RNA found all living organisms, including bacteria, virus, plants, animal. The all living organisms here. And this nucleic acid function as the structural molecules in the cell organelles and are also involved in the catalyst of biochemical reaction here. That's important. The different types of RNA are involved in the various cellular process. Let's see. First of all here, facilitated of transcription of DNA into the protein. That's important. Also show in this picture here of facilitated their translations of DNA into protein and function as the adapter molecules in protein synthesis and serve as a messenger between the DNA and the ribosome, and they are a carrier of the genetic information in all living cell and promotes the ribosome to be choose the right amino acid which is required in a build up the new protein in the body. That's important functions here. The next here, types of RNA. That's important types of RNA here. You know that first here types tRNA, transfer RNA, ribosomal RNA, and third here messenger RNA. You know that three types of RNA here. First of all here transfer RNA, the tRNA. The transfer RNA is held responsible for the choosing the correct protein. That is choosing the correct protein or the amino acid and required by the body in turn helping to the ribosome. That's important transfer RNA here. And ribosomal RNA means rRNA here. There's a functions here in all living cell. The ribosomal RNA play a fundamental role in the synthesis and translation of the mRNA into the protein. That's important here. The ribosomal RNA is all living cells present here. The ribosomal RNA play as a fundamental role in their synthesis and translations of mRNA into proteins. And the next here, messenger RNA. Messenger RNA is the mRNA. That's important. The mRNA play a vital role in the process of transcriptions of during their protein synthesis process. That's important here. Messenger RNA play a vital role in the process of transcriptions of during their protein synthesis process here. But next question arrive here, that's important. During which stage can be chromosome be seen clearly? You know that the question is clear here. During which stage can be chromosome be seen clearly? Write the feature of eukaryotic and prokaryotic chromosome. You know that that's important. Question for the exam view here. There is a chromosome be seen clearly and some feature of eukaryotic and prokaryotic chromosome. First of all here, chromosomes are distinctly visible during to the metaphase stage. Metaphase stage or mitosis of a cell. You know that the chromosome, there is a clear scene here, metaphase stage here of mitosis of the cell division. And let's see feature of eukaryotic and prokaryotic chromosome are following here. First of all here, eukaryotic chromosome here. You know that that's important. Eukaryotic chromosome here. They are present in a large numbers. First of all here, eukaryotic chromosome, they are present in a large numbers. In a higher organism, it occurs in the paired condition. You know that in a higher organism, it is occur in a paired condition here. And the next here, they are visible distinctly only during to the metaphase stage of mutasis. That's important here. They are visible distinctly only during the metaphase stage of mutasis. And the next here, each chromosome consists of two chromatics attached together by a centrome. That's important. Each chromosome consists of two chromatics attached together by a centromere. That's important. And let's see a eukaryotic chromosome here. The next eukaryotic chromosome here, that's important. They are a simple inner composition. They are a simple inner composition when the compare it to their eukaryotic chromosome. And the next, they are generally circular in a shape. They are generally circular in a shape, and there is only one chromosome in a cell. That's important here. There is only one chromosome in a cell. And the next question arrive here, that's important. Define a genetics. Define a genetics, and what is the contribution of the Mendel in the field of genetics? You know that is an important question here. Define the genetics and what is the contribution of Mendel in the field of genetics. First of all here, the branch of biology that deal with the study of heredity and variation now as a genetics. You know that is important. The branch of biology that deal with the study of heredity and variation known as the genetics. You know that what is genetics? Genetics of branch of biology deal with the study of heredity variation is known as a genetics. George Jones Models was the first person to carry out experiment regarding the heredity of content of character from one generation to another in a scientific manner. That's important. George Jonas is Mendel, the first person who is the experiment regarding the heredity and contain the character from one generation to another in a scientific manner. He worked mainly on the garden plant. You know that the Mendel's is a select here a garden pea plant. His observation regarding their occurrence of contrasting character in various generations of garden and pea lead him to the interrupt that these are controlled by the unit which he is called factor, and this factor are today known as a gene. This factor, who factor? There is a character of various generation of garden pea lead him to interrupt that these are controlled by the unit which is called factor, and this factor is today known as the genes, and he is also known as the father of genetics. There is the next question arrive here, that's important. What is a sex chromosome and types of chromosome? You know that that's an important question here. What is a sex chromosome and here are types. The chromosomes that are responsible for the determination of sex in an individual are known as the sex chromosome. You know that is important. Chromosomes that are responsible for a determination of sex in individual are known as the sex chromosome. There are two types of chromosome here, X chromosome and Y chromosome. That's generally two types here, X chromosome and Y chromosome. The male possesses one X and one Y chromosome here, but female possesses two X chromosome. You know that's important. The male here opposes a X and Y chromosome, and other side females opposes two chromosome, XX chromosome. That's important here. And let's see types of chromosome generally here. The types of chromosome, that's important. There is a centromeres here. You know the types of centromeres here. That's important. The first centromeres here, metacentric. First of all here, metacentric. It is a chromosome with the centromere near the middle and two chromatics of equal length. You know that this metacentric is a chromatics are equal length. And second here, submetacentric. Submetacentric here, the centromere is situated is slightly closer to one end than the other. That's important. There is a centromere is situated slightly closer to one end than the other. And thus one chromatic is slightly longer than to the other. That's important point here. And the next here, acrocentric. Acrocentric, here the chromosome is situated near one end of the chromosome, and thus one achromatic is very long while the other is very small. You know that the chromatic one here is very long and other is very small. And telocentric here. Telocentric here, the centromere is situated at the tip of the chromosome. You know that is important. And let's see here are centromeres on figure here. You know that on your screen there is a centromere here. Centromere is a 4 types here, generally explained here. And the centromeres, there are first of all telocentrics. I have already described there are telocentrics here. And second is a acrocentrics here. And third, submetacentric here. And last is a metacentric. You know that is important. And the upper side is a P arm and lower side is a Q arm. That's important. And centromere here, a center of part. This is a 2 circle, the show of centromeres here. That's important diagram here. How is a sex determination in human being? That's important question here. How is a sex determination in human being? The male sex gamete have a X and Y chromosome. The male sex gametes have a X and Y chromosome, whereas the female sex gametes have a 2 X chromosome. Whereas sperm containing a Y chromosome fused with the ovum contain a X chromosome, the zygote develop into the male. That's important here. The female gamete have a 2 and there is a when sperm contain a Y chromosome fused with the ovum contain to the X chromosome. There is mother X chromosome, there is zygote develop into the male. And when a sperm contain the X chromosome fused with the ovum or contain X chromosome, the zygote develop into the female. That's important here. And thus the sex and the individual is determined by the sex chromosome X and Y, which is present in the male chromosome. That's important. X and Y chromosome present in the only male chromosome. That's important. The boy and the girl depended here a father gamete. That's important point for the exam view. And let's see step by step for this processing. Let's see step by step on this processing. There is the humans have a 23 pairs of chromosome. And out of these 23 pairs, 22 pairs are autosome and only 1 pair is there a sex chromosome. That's important. Out of these 23 pairs, but 22 pairs is the autosome and only 1%, 1 pair is the sex chromosome, and which actively take part in the process of sex determination. And the next, both male and female carry 2 sets of sex chromosome. Both male and female carry 2 sets of sex chromosome here. And male have 1 X and 1 Y, means XY sex chromosome, in which both are the active. Males here X and Y chromosome and the two chromosomes here, and both are active. And other side, female has a both X chromosome, means XX sex chromosome, in which one is active only. There is a one active only. That's important. The XY sex determination system is found in a human. That's important. XY sex determination found in human, but not a human. There is a mammals and some insect and few plant species also. That's important. And all children will be inherit a X chromosome from their mother. The all children, male and female, there is a X chromosome for the mother, and that there are whether they are boys or girl, and thus the sex of the children will be determined by the types of chromosome inherit from their father. The father is the main suspected here, dominated by the X and Y chromosome here. A child who is inherit Y chromosome will be a boy, and who inherit X chromosome will be a girl. There is a Y chromosome is here a boy, and the X chromosome donated by there is girl, and sex determination boy or girl. That's important. But first of all, most important point here, a male semen has a whole approximately 50% of Y sperm cells, and other is a 50% of X sperm cell. You know that is important. And the boy and the girl, the 50-50 chances here. The male semens hold approximately 50% of Y chromosome and 50% of X chromosome sperm here. And therefore there are a possibility that a man have a 3 or more children of a single gender. You know that is important. The X and the here is important. And the let's see diagram here. Here show the diagram on screen. That's important for the exam view. Sex determination in human being. That's important. You know that there is a parents here, mother and father, and gametes, reproductive cells here, and the last here a zygote formed after fusion here. You know that father is donated for there is the male X chromosome, produce a female, and the father here again X chromosome is a mother, there is a female, but other side father is a Y donated for the mother X chromosome. In this condition, the male child formations, and other side Y chromosome and mother X chromosome, there is a male child here. That's important diagram here. It means to 50-50 chance for the girls and the boy. That's important. Next question arrive here. That's important. Explain how the sexual reproduction give a rise to be more variable variation than the asexual reproduction. You know that is important. The question is clear here. Asexual reproductions and asexual reproduction. But here sexual reproduction give a rise to more variable variation than asexual reproduction. You know that this is the most important feature here, sexual reproduction here. Because of the following factors, sexual reproduction lead to be numerous variable variation here. That's important. First of all here, a mistake in DNA copying. That's important. The mistake in a DNA copying through its was rare. That's rare here. And at the moment, sex cell develop a paternal and maternal chromosome are randomly segregated. That's important here. The sex cell development, paternal and maternal chromosome are randomly segregated. That's important. When the gametes are formed, homologous chromosome and exchange the genetic material. That's important. When the gametes are formed, homologous chromosome and exchange the genetic material. That's important. The next year, the variation accumulated as a result of reproduction over many generations and extensive diversity and natural produced by the choose. That's important here. Variation is accumulated as a result of reproduction over many generations and extensive diversities as naturally produced by the choose. Because there is only one parents engaged in the asexual reproduction. Because there are only one parents engaged in a asexual reproduction. variation in highly is a constrained. As a result, the offspring share the parents' genetical makeup. That's important here. Genetical makeup is of share. Next question arrive here. That's the most important. Explain a man does experiment with the piece on the inheritance of the character, considering only one visible contrasting character. You know that is important. That's important for the exam view here. Explain the man does experiment with the P on the inheritance of their characters, considering only one visible contrasting character. You know that is important. The pure tail pea plant and the pure draft pea plant were crossed by the Mendel. Every F1 generation plant harvested was tall. Every F1 generation plant harvested was tall. Mendel obtained both tail and draft plant in the ratio of 3:1 in the F2 generation. When the cell plant from the F1 generation, that's important experiment Mendel's here, and this explained by why there are two alleles for each set of the opposing characteristics. You know that is important. This mentions why there are two alleles for the each set of the opposing characteristics. A dominated alleles control the trait that is expressed in the first generation, means F1, whereas the received genes regulate the trait that is not expressed in the first generation. That's important here. A dominant alleles control their traits that is expressed in their first generation F1, and whereas the receives our genes regulate their traits that is not expressed in the first generation. That's important. And when the both opposing alleles are present in the F1 individual, and there is no mixing of alleles. Instead, there is segregate once or more during their production of the gametes. That's important here. Instead, there are segregate once or more during their production of the gametes. Received stress, therefore, this surface is their F2 generation. As a result, the combination of the received genes. You know that's important. Received stress, therefore, this surface in the F2 generation as a result of the combination of the receiver genes. That's important here. And let's see 3:1 ratio diagrams here. You know that on your screen there is a most important here monohybrid cross here. There is a monohybrid cross here. A trace seed shape and the alleles R is round and R brinklet. That's important. And cross here round seeds into round seeds. That's important here. Cross here. There is this capital R and cross here capital R and R. That's important on your table. That is an important character 3:1 ratio here. The RR is the first here. That's important. A genotypes is RR and the R small r and small r here. A phenotypes is a round and brinkling. Means a G ratio is a 1:1. There is a 1:1. But it is genotype ratio is a 1:1 and phenotypes is a ratio here 3:1. That's important here for the exam view. Know that is important. The genotypes ratio here 1:1 and phenotype ratio is 3:1. That's important. Give the basic feature of the mechanism of inheritance. That's important. Flowings are some of the fundamental components of the inheritance mechanism here. First of all here, characters are controlled by genes. Characters are controlled by the genes. That's important point here for the exam view. Characters are controlled by the genes. Genes cannot take two or more different form. You know that genes can take two or more different form here. Genes are found on chromosome and one type of gene may be a dominant over to another. That's important. Genes are found on chromosome, and one types of gene may be dominant over to another. There are two possible versions of single gene, which may or may not be a similar. There are two possible versions of single genes which may or may not be a similar. At the time of gamete creation, the gene split into two forms. At the time of gamete creation, the genes split into two forms. In the zygote, two genes from a come together. In the zygote, two genes from a come together. That's important. Next question arrive here. That's important. How do the Mendele's experience show that our traits may be either dominant and receives? That's an important question here for the exam view. A Mendele's experiment showed that a trait may be either dominant and receivers here. You know that Mendele's test on the monohybrid cross and showed that a trait might to either to the dominant or receives. That's important. And he conducted the experiment by the crossing of tall TT means capital T and draft here a small t and the use the peace plants here that's important. There is a conduct experiment by the crossing here tall and draft. The F1 means of phyllos is the generation was represented by the plant that emerged as a result of the fertilization. Means the F1 generation here. The F1 year plants were all very tall. You know that the F1 plants all very tall. That's important experiment here. Mendel continued by the cell pollinating the plants from to phyllos generation and a result 1/4 of the plant produced by the F2 generation were a draft. That's important here. Mendel continued to sell pollinating their plant from the phylls generation. As a result, 1/4 of their plant produced by the F2 generation were a draft. Mendel deduced from to the experiment that the tall F1 plant were not a true breeding. You know that is important. Mendel deduced from the experiment that the tall F1 plants were not truly breeding here. Instead, there is a positive trait of both tall and dwarf heights because the characteristics for the tallness as a predominated over trait from the dwarfness. Some of the plants were tall. That is important. The idea that the traits can be either be a dominant and receives in thus the confined here. That's important. Let's see a hybrid diagram here. On your screen, there is a most important diagram here. Monohybrid cross. You know that monohybrid cross is the most important. Here show the F1 gametes means first generation a capital T and a small t. Another side here capital T and a small t. You know that F1 gametes is a present here a tall and tall tree. You mean first generation is old for the tall. But come to the F2 gamete is a second generation. That's important. There is a first of tall tree but other is a short means draft mean TT here. Is a capital T is a tall plant is a show here and D is a draft of plant. That's important for the exam view and ratio obtained here 3:1. That's important. There is a 3:1 phenotype type and genotypes here. You know that next question arrive here. That's important. How do the Mendel's experiments show that a trait and inherit independently? You know that that's important question for the exam view here. Mendel's is the hybrid cross experiment demonstrated that his characteristics are inherited separately here. He used two characters in the experiment. There is used here two character sticks in the experiment here. First here, seed shape and seed color. Seed shape and seed color, that's important here. Well, the round shape is RR, is a predominant here. And over that brinklet shape, there is color yellow, means capital YY. And dominated over green here, a small y and a small r. That's important experiment here. There is a 9 plant with the round yellow, means capital R and capital Y seeds, and 3 plants with a round green, means capital R and small y. 3 plants with a brinklet yellow, means small r and capital R here. 1 plant with a brinklet green seed among the F2 progenies of the hybrid cross here, a small r and a small y. The ratio here 9, 3, 3, 1. That's important here. 9 ratio 3, 3 ratio 3 and ratio 1. That's important here because there is a 9 plant with a round yellow shape and 3 plants around green and 3 brinkling yellow and the hybrid cross here 1. That's important experiment for the medlefs here doing here. He also noted that there is a round green and brinklet yellow are new. The brinklet greens and the round yellow are parental combination here. That's important. That's important for the exam view here. The round green and brinkling yellow are new. The brinkle greens are rounded yellow are parental combination here. For different types of gamete were produced by the hybrid cross between two seed with the dominated stress capital R and capital Y, and non-dominated trace is a small r, a small y. That's mean ry is r, a small y, and the r capital Y and the r. That's important for the exam view here. By disaggregating independently of one another, each gamete occur at a frequency of 25% of total number of gametes generated. That's important. Let's see diagram. That's important for the exam view next year. On your screen, there is a most important the hybrid cross here. You know that is important on clear on your mind. There is a pure breeding round yellow seeds here, capital R and capital Y, and pure breeding brinkling green seeds, small r, small y here. You know that there is a cross here of parents and parents gametes, capital R and Y, and a small r and a small y. You know that first generations here, round yellow seed is a pollination. That's important. And here a table on your clear, there is a pure breed has a yellow here, a 9, yellow round here 9, and yellow brinklet is 3, and green round here 3, but one green brinklet is 1. On screen you're clear on second generation F2 here. Know that is important for the exam view. That's important here. And our last question arrive here, that is the most important here. That is the most important for the exam view. Examiner many times asked in these questions here. Why did the Mendel select the pea plants for the experiment? That's important question here. Why did Mendel select the pea plant for his experiment? You know that Mendel experiment is the most important. First of all, Orizors Johannes Mendel is also known as the father of genetics. Who is the father of genetics? Is a G.J. Mendel. That's important here. He has studied the inheritance of 7 pairs of contrasting traits present in the garden pea plants here. That's important experiment Mendel's here. He proposed three principles of heredity based on this result. You know that he proposed the three principles of heredity based on this studies here. You know that the reason why the Mendel chose the garden peas plant for the experiment. That's important question here. You know that first of all presence of observable trait with the contrasting form. First of all reasons here, presence of observable trait with the contrasting forms here. And second here, it's a produce many offspring in one cross. It is important. It's a process is a many offspring in one cross here. And they are easy to the cultivate. They are easy to cultivate here, and their life cycle is quite short. That's important here. The life cycle is the quite short. The flower of pea plants are bisexual. This is important. The flowers of pea plant are bisexual here. That's important. And the next here, beside the performing self pollination, that's important. There is a performing here self pollination, and this plant can undergoes to crow pollinations if it is a perform one them. The true breeding or pure plants can be easily obtained through the self pollination here. That's the selection reasons here, and their characteristics can be distinguished properly without any difficulty. This means that a trace in the plant can be easily absorbed. There is a easily absorbed, and this plant supposes a large flowers, and therefore anthers is a male can be easily removed to the make this flower cross with the pollens from anther flower. That's reason is a select to the middle of is a pea plants here. That's important for the exam view here. This topic was till this. We will be do their next topic in the next part. If you like it, then please like, share, subscribe, and share the video as a much as a possible. Do not think subscribe and save in your mobile.